welcome back, everyone. Thank you again for attending the ORC IAP seminar. And I have the pleasure of introducing Professor Uhler. Professor Uhler joined MIT in 2015 and is currently the Henry L. and Grace Doherty Associate Professor in EECS and IDSS. Her research focuses on mathematical statistics and computational biology.
Thank you very much, and thanks for the kind introduction, and in particular also for inviting me to speak here. So you'll hear the second talk on causal inference today. Well, Nathan's talk was mainly on observational data. I am particularly excited to be working on causal inference right now because for the first time ever, we actually have access to very large-scale interventional data. And until now, basically it has been either unethical or impractical or just extremely expensive to perform randomized experiments. Through the development of the CRISPR-Cas system in genomics, it's for the first time possible that basically anyone can perform really large-scale interventional data or interventions. And now, since maybe a couple of years ago, say 5 or 10 years ago, the genomics data sets were maybe like 100 observations on 20,000 variables, so that's how many genes we have in humans. Today, all of the data sets are like 100,000 to 1 million observations on single cells over all 20,000 genes. And not just can we intervene on any one of the nodes, but we can intervene on any combination of nodes. And basically any undergrad can perform these interventions. So I think this not only allows us to test, of course, all of the causal inference algorithms that have been developed, but it also leads to completely new questions, right? How do we actually do causal inference? How do we use this data? How do we now do causal inference based on a mix of observational and interventional data? So these are the kinds of questions that we will be talking about. And I'm taking a graphical models approach, so very different to Nathan's approach this morning. Graphical models, just because if I want to do something on 20,000 nodes, I need to be able to represent it. Okay, so I cannot ask, or in these kinds of questions, you're not interested in just asking about one particular causal edge. Is there a causal relationship or not? I really want to be able to say something about all of these nodes that are there in this network. So I'm sure you all know how graphical models are used all over the place because very often you have like these huge data sets and you want to be able to say something about the relationships among different variables. And so graphical models, they represent this by a network, which not only enhances interpretability, but often also leads to computationally efficient ways of actually analyzing the data. And so depending on the application, you might want to use undirected graphical models, directed graphical models, et cetera. So for example, okay, so this doesn't work. So for example, in weather forecasting, anyways, you cannot intervene on the weather. So usually, people just use undirected graphical models. However, if you have genomics or medical applications, you really need to know the causal relationships, right? I want to know what happens if I intervene on a particular node. Which genes are actually downstream? Where does something change? Which genes are upstream where nothing is going to change? So a lot of what we do is going this extra step from predictive modeling, where there's quite a bit of research all the way over to actually causal modeling. And okay, so this is a machine learning day. And we all know, in machine learning, you often don't want to make modeling assumptions, right? You try to just directly optimize prediction performance. And this has led to spectacular results, as we all know. But for some applications, this is just not enough, right? So just prediction is often not enough, in particular in medicine, where you need to know something about the underlying mechanism. You need to know the underlying causal mechanism. And so this is about really kind of opening up this black box and really saying something about this underlying causal mechanism. And I think machine learning, because it tries to optimize prediction performance, it's really kind of geared towards analyzing observational data. And so the question is, how do we actually make use of interventional data? And so an overarching goal of our research is really to develop a theoretical and algorithmic framework for being able to infer these underlying causal mechanisms from a mix of observational and interventional data, right? Because if you don't have interventions, you can in general, and not even, Nathan this morning was talking about when you have unobserved confounders. But even if you don't have any unobserved confounders, in general, if you just have observational data, you can in fact not infer the underlying causal mechanism. You really need interventions. So the question is how to do this. So obviously, I should also say a bit about the history because, causal inference is the cornerstone of scientific discovery. So obviously, it has a long history, right? And the framework was already developed in the 1920s the framework which we're going to use is these structural equation models. They were introduced in the 1920s by Jerzy Neiman and Sewell Wright. But then afterwards, something happened that, like, for 50 years, basically, there was a lot of skepticism among statisticians, actually, about this framework. And this basically halted the whole development. And only very recently, in the 1970s, causal inference came up again. And this was all from observational data, really based on, very big contributions from a variety of different people, namely Julia Pearl, Jamie Robbins, Don Rubin, and Peter Spertus. And I put the areas here just so that you see how interdisciplinary this area of causal inference is. And you'll see a bit of it afterwards when we talk about the different kinds of methods that come in. But I think really now is the time to make big leaps, and I already said this, because really for the first time ever, we have all this interventional data, okay? And the kind of data I'm thinking about is genomics data. So think about it in this way. You have 20,000 variables, and you have 100,000 to 1 million observations. And these can be observational or interventional. You can intervene on any one of your nodes or any combination of nodes. Okay, now a cell dies if you make too many interventions at the same time. So usually you don't make more than 5 interventions at the same time. Okay, but still, 20,000 choose 5 is a pretty large number, right? So there are many new questions. In particular, how do we even deal with, like, what can we learn from interventional data? How do we use it? Okay, so let me set up the statistical framework that we're going to use. So as I said, it is structural equation models that were already introduced in the 1920s. So since I'm talking about modeling causality, you can never cause something in the past. So here we will assume that it is a directed acyclic graph. Okay, so your causal structure, your underlying causal structure can be modeled as a directed acyclic graph. And every node is a random variable, so think of this as genes. So you get observations on the genes. You would like to say something about the underlying causal mechanisms. Every node is some function, deterministic function, of all of its direct parents and some independent noise. Okay, I'm making no assumptions on what these forms are, so these functions can be anything. They can also be nonlinear, etc. I'm also not wanting to make any parametric assumptions on the noise. I'm not saying it's Gaussian or anything like that. This is the model that I'm going to use. Okay, so this is the most general structural equation model, and this is the kind of model that I would like to learn. Okay. Now, if you're familiar with Bayesian networks, and I assume everyone here is familiar, if I would stop here, then this is basically not causal, right? This is just modeling the observational distribution. What makes the difference to a structural equation model is that it also encodes all interventional distributions. Okay, so I need to actually set up how I'm talking about, or the framework for talking about interventions. And so I will distinguish, based on Frederick Eberhardt's work, I will distinguish, sorry, this really doesn't work. I will distinguish 2 different kinds of interventions. The most invasive intervention is known as a perfect intervention. So what is that? So a perfect intervention on some node is you just set that node to a particular value. Okay, so if I'm thinking of gene expression, I'm just deleting a gene. I'm doing a gene knockout experiment. I'm just setting it to 0. Okay, this is a super invasive intervention. I'm even changing the network structure, right? I have no more effect of my parents, of the causal parents on me, if I'm just setting my value to 0, for example, right? So I'm just removing all causal edges that point into me when I do something like that. Oh, thank you so much. This is great. So that's a very invasive intervention. And everything else is called a general intervention. So here in a general intervention, maybe I'm just doing something wrong. But do you guys see something? Or you do? It's not. Oh, OK. So maybe it's just like how I push buttons or something. Oh, great. Thank you so much. OK. Woo, great. So in a general intervention, I am just, for example, changing how the causal parents act on me, or I am changing the noise that I'm introducing in each one of the variables. Okay, so there is a big difference. So here I'm just making changes to the causal effects, and here I'm really setting the value of a particular node somehow deterministically, so that means that I'm removing all of these causal effects. Okay, and so then there are many basic questions that we started asking. Like, for example, from a biological perspective, well, they try to do gene knockout experiments because it seems that you should gain more causal information from a more invasive experiment than from a less invasive experiment. So we asked if and what kind of causal information can you gain from a perfect intervention that you cannot gain from an imperfect intervention? And then, of course, all the algorithmic questions, right? How do you actually learn this network from a mix of observational and interventional data? So these are the two questions that I want to answer in this talk. OK, so I also have to say a bit about the standard approaches for learning these underlying causal networks. So the standard approach is, well, I have a structural equation model. A structural equation model implies certain constraints on the distributions. Right? In particular, if you have certain missing edges, that implies certain conditional independence relations. And these are just the simple ones, OK? So these are the ones, a node is independent of its non-descendants given its parents because any causal or any information that this node has on its non-dependents, on its non-descendants is already, it has to go through the parents, and hence if you're conditioning on them, then nothing goes through them. Okay, and in fact, these are the only ones you have to know because everything is already implied by these kinds of simple conditional independence relations. So the standard approaches is, OK, I have my data. I learn all the conditional independence relations that hold in my data. And now I try to go from conditional independence relations to my network, OK, by just going backwards. So a network implies certain conditional independence relations. Now I just want to go the other way around. Now there is a problem with this, and you all know this if you know Bayesian networks. There are different models or different graphs that encode the same conditional independence relations. So just from observational data alone, you cannot even hope to learn the full underlying causal structure. Okay? So you can only learn a Markov equivalence class. And here, just as examples, right? So these are all the equivalence classes on 3 nodes. So for example, if you just have observational data, I cannot tell if x causes y or y causes x. Right? I just get that they're correlated. I really need to do an intervention on these nodes. And so obviously, then there are all these questions, right? Now that I have interventional data, well, how much? So identifiability increases, but how much more? Right? So what are the equivalence classes when I actually have, for example, here, say I have an intervention on x. How do these interventional equivalence classes change? Right? How do these break up? Or, and this is the question that I was asking at the beginning, now framed mathematically, if I have perfect or imperfect interventions, do I get the same interventional Markov equivalence classes? Okay, so do they provide me, at least in the limit, when I have an infinite amount of data, do they provide me the same amount of causal information? Okay. So that's kind of how we're framing, all these questions about interventions in a mathematical manner. So before I can talk about interventions, let me just give an overview on the kinds of algorithms that are around and that have been around because, studying causality from observations is a very old field, as we've seen. Okay, so I'll just give a very brief overview on the different types of algorithms that have been around in the observational setting, and then I'll get back to these questions in the interventional setting. So what are the algorithms? So it's actually kind of nice because it's very clean. So in the observational setting, there are basically just 2 different approaches, okay, for learning the underlying Markov equivalence class from observational data. One is constraint-based and one is score-based. And I do want to say a bit about what they are, and just so that we have an understanding of what are the advantages and disadvantages if you actually want to use some of these algorithms. So what are constraint-based approaches? Well, you have data. As we said, we'll go to conditional independence relations. And what they do is they try to find a network that satisfies as many of these conditional independence relations as possible. Okay? So these are the constraints, the conditional independence relations. Now, you already see a disadvantage, right? Because constraint-based approaches, if you don't get the constraints right, well, you're definitely not going to get the network right. But your constraints are learned from data. So you probably need quite a lot of data in order to get the constraints right so that your algorithm will actually perform pretty well. Okay? So you always have this trade-off in statistics, right, between computational costs and statistical, how many samples you need. This is an algorithm which is extremely fast, or a class of algorithms that are extremely fast, but you need quite a lot of data in order to get a good outcome. Then we have something on the other side, which are these score-based approaches. So what do they do? So you have a score, usually a penalized likelihood score, and now you just do greedy search over your space of, say, Markov equivalence classes. Okay, so now you have the other problem. Well, this space, and we'll see it on the next slide, is huge. So you have to do greedy search on a huge space, so that's usually slower. But at the same time, because it's greedy, you usually do much better with smaller amounts of data. Okay, so you are on this curve, of statistical computational trade-offs, you're on the other side. Okay, so more costs from a computational perspective, but better in terms of statistical properties that you get out. Okay, and then there are a lot of mixes in between these algorithms, but these are the two approaches for actually doing, causal inference when you just have observational data. Okay? And on the next, I want to give you just a bit more intuition and some open problems about these, advantages and disadvantages of these two algorithms. And I'll start with score-based. I said, the search space is huge. Well, let me go a bit deeper into this. So we have to ask ourselves, how many Markov equivalence classes are there? Right? Because already I'm not doing search over DAGs, I'm doing search over a smaller space, so maybe that's much smaller than the number of DAGs. So this problem is actually not known, so that's the first open problem. So it's not known if I give you P, or N in this case, vertices, how many Markov equivalence classes are there? Okay, it's known how many DAGs there are, how many possible DAGs, but not how many Markov equivalence classes there are. So Gillespie and Perlman have a very nice paper where they just enumerate them all, up to 10 nodes. Okay? And it seems like it really explodes very, very quickly. So it's here for 10 nodes, it's already 10 to the 19. So this is a pretty nice combinatorics question. But what I really like are these two columns. Okay, so these are important. So what is this? So this is the total number of Markov equivalence classes divided by the number of DAGs. So they say acyclic directed graphs. So these are DAGs. Okay, so this seems, and again, this is a conjecture, that it seems to converge to something like a quarter. Okay, so this says that in average, every Markov equivalence class contains 4 DAGs. So this is if this is correct, that's really important, right, if you think of interventional data, because it tells you that in average, you don't need that many different interventions in order to be able to pinpoint, what actually your true DAG is or your true causal network is. Right? So this would be another very important kind of question to actually prove results like this. Also, this is very interesting. This is the total number of equivalence classes of size 1 divided by the total number of Markov equivalence classes. Okay, so these are how many, and it seems to, again, converge to something like a quarter. So this is how often, when you're running these algorithms, you actually get a unique DAG out. So you can already identify your graph just from observational data alone. So it seems that in about a quarter of the cases, you actually don't even need interventional data in order to be able to identify your underlying causal network. But these are all conjectures, okay? So what we looked at in these papers is, we proved some hardness results. We looked at this also for certain classes of random graph models. But here, I think there's still a lot of open questions to actually get to this, like to really answer these kinds of questions of these convergences, et cetera. Okay, to really know something about how many interventions do we need in order to identify the underlying causal mechanisms. Okay, so this tells you a bit about why these algorithms, these score-based approaches are in general slow. Okay, the search space is just huge, or seems to be huge, although we don't have the correct number here. So it basically seems to be up to a constant equal to the number of DAGs. Okay. And maybe also something about the limitations of the other algorithms, the constraint-based algorithms, and why they need so much more data. So as I said, right, so the problem is really that if you don't get the conditional independence relations correct, well, how they work is, if I have a conditional independence relation, I'm just going to remove an edge. So that requires, and here you see the influence of philosophy, this requires something known as the faithfulness assumption. So the distribution should be faithful to your graph. Okay, so you shouldn't have conditional independence relations that are spurious, that are not already given by your graph, because otherwise I'm going to remove an edge that I shouldn't have been removing. Okay? And so why is this a strong assumption? So let me just go over this with an example. So say you have like some medicine, which has like some positive effect on your lung functioning. Okay? But it also has a negative effect on your immune system. And obviously, if you have a well-functioning immune system, it will have a positive effect on your lung functioning. So it could happen that these causal effects just cancel each other out. Right? If they cancel each other out, well, then you get that these two variables are independent. Hence, you're getting like a wrong your algorithm is going to make a mistake because it's going to remove this edge, and in addition, get all kinds of directions of the edges wrong, right? So that's a faithfulness violation. But now you can tell me, well, this happens with probability 0, right? That these causal effects directly cancel each other out. Well, that has measure 0. And this was exactly also the argument they gave for actually making that assumption. Okay? So the problem is that you're working with data, right? So these were in philosophy, but we're now working with data, so we should take a statistical approach. So even if the true model has an independence relation, right, your data is not going to be exactly independent. You have to perform a test. You have to introduce some threshold, which depends on your sample size, right? And so you don't need just faithfulness assumption, but you need something stronger, right? Basically, you need to fatten up this faithfulness assumption. And so we try to make this quantitative, like how bad is this assumption really? And here I'm just going to show it to you on a very simple graph and very simple model. Okay, so say this is the truth. And I'm just taking a linear structural equation model with Gaussian noise. Okay, so all of my causal effects are linear. And I'm just doing the simplest model possible, just Gaussian noise. Okay, and I only have 3 variables so that I can actually draw everything in 3D. Okay, I just want to be able to show you what is happening. So what is, and we don't have to go through all the math, but so what are all unfaithful distributions? Well, they're any distribution that satisfies some conditional independence relation, right? Because this is the complete graph, so I shouldn't have any conditional independence relations. And now believe me, right, you can go through the math. In the Gaussian setting, any conditional independence relation can just be written out in terms of the covariance matrix. Right, you can just do the math. I have 3 variables. I'm just solving. And so I get all of these polynomial equations. So whenever your causal effects satisfy any of these equations, well, then I'm going to remove an edge that I shouldn't be. Okay, and now I can actually draw it. And I can tell you that the algorithm is going to make a mistake whenever you're on any of these hypersurfaces. Okay, but not just exactly on the hypersurface. If you don't have infinite data, well, I have to fatten up these hypersurfaces. Right? Like 1 over square root of n is your threshold that you have to use. And so you see, and now this is a small example, but you see that if I'm starting to fatten up these hypersurfaces, very quickly they're going to fill up the whole space. Okay, and that's exactly the kind of math we did. And we're able to figure out, how many variables are you actually allowed to have given, a given sample size that you have, so that this algorithm has any chance of getting anything right. Okay, so this is the problem. So for these constraint-based approaches, you just need a huge amount of samples when you have, some pretty small number of variables, okay, to have any hope that you're actually going to get the correct. And that's just because of this problem here, that you're just at some point going to make a mistake in terms of your conditional independence relations. Okay, so we have these 2 extremes, and again, since this is for a lot of PhD students, just pointing out a lot of open questions. So I think what is completely open and has been studied for other statistical problems, in causal inference, we don't know this optimal curve of statistical computational trade-off. Okay, I'm telling you, we have these 2 algorithms. They're somewhere, one is here and somewhere here, but we have no idea what this optimal curve is. And do these algorithms actually lie on the curve or not? I would think not. But we actually don't know what this is. Okay, so that's a big, big open question, I think. And so we try to at least, we have 2 points. Let me at least try to find the 3rd point with a different algorithm somewhere on this curve here, which hopefully is better in terms of computation time as these score-based approaches that we had, and hopefully also better in terms of statistical properties than this constraint-based approaches that we had. Okay, and that's something that we developed and also led to all of our algorithms that we were able to develop in the interventional setting. They're all based on this, and that's why I wanted to just briefly go into it and tell you what the main idea is behind this new types of algorithms that we have. So the main idea is the following. So if you want to learn a DAG, okay, any DAG, I'm going to say something, I don't know, something like this, something simple. I'm going to represent this problem as 2 separate problems. Namely, one is learning the skeleton, so learning the undirected graph. And the other one is learning the ordering of the variables. Okay, so in this case, I would give you this, and I'm telling you that 1 is smaller than 2 is smaller than 3. Well, that tells you that I should orient my edges in this direction and in this direction. Okay, so instead of learning a DAG, I'm going to say I just need to learn an ordering, so the permutation. And we all know that learning undirected graphs is pretty easy. So if I give you an ordering, you can actually associate to it the undirected graph, and this is well studied. So I'm going to leave that problem out, okay? This has been studied, for example, already by Julia Pearl very early on, so I'm not going to talk about that. So I'm going to phrase the problem of learning a DAG just as learning an ordering. Okay? So instead of greedy search on space of DAGs, etc., I'm going to do greedy search on space of orderings. Okay? So this is how you associate to any, and let's not go into it, how you associate, this has been defined by Julia Pearl, how you associate to any ordering an undirected graph. Okay, so this is easy. So all I need to tell you is how do we perform? So now for every permutation, okay, this is the space of permutation, I can associate to it an undirected graph or a DAG. Okay, so I have to tell you, I'm going to do greedy search. I have to give you a score function, and I have to tell you what it means to do greedy search in the space of permutations, which probably everyone in here knows. Okay? Score function, we want it to be non-parametric. I don't want to use some, penalized likelihood function. I don't want to do Gaussian assumption, in particular not for gene expression data. So we're just going to use number of edges in the graph. Okay, the simplest score function you could come up with, just the sparsity of the graph. So I'm starting in some permutation, okay? Now one thing is space of permutations is a polytope, right? It's known as the permutahedron. So I have to tell you what it means to do greedy search on the space of permutations. So if I give you any permutation, this is a polytope, my neighbors are in fact known. So my neighbors are given by neighboring transpositions, okay? So most people are nodding, so they know this. So if I'm here, for example, in this permutation 3, 2, 1, 4, my neighbors are, where I can transpose the first two elements, I get to 2, 3, 1, 4. I can transpose to 1, I get to 3, 1, 2, 4, or I can transpose the next two guys, I get to this guy. Okay, and this is true for any dimension. So the space of permutations is a polytope, and I know who my neighbors are. So I can actually run a simplex type algorithm. Okay, so I gave you a score function, just the number of edges. You start in any permutation, you compute its undirected graph, you count how many edges you have, you look at your neighbors, and you just walk there if you have less edges. Okay, that's a super simple algorithm, and in fact, we can prove that this is consistent under strictly weaker conditions than both of the other types of algorithms that were around. Okay, so you have a smaller search space. In addition, you also get consistency under strictly weaker conditions. Okay, so just by phrasing somehow this problem as a problem over permutations, you get out all of these things. And just to show you how this algorithm works, I mean, this is just a little nice math, how you get into like really nice math when you work over permutations. So if I have a graph like this, for example, right? So this graph is consistent with the ordering 1, 2, 3, but obviously it's also consistent with the ordering 1, 3, 2. So I already know if I'm sitting in this ordering, I already know that if I go to this ordering, I'm just going to get the same graph out. So I already know that the score function is not going to change. So I shouldn't be searching over the whole space of permutations. I can actually search over a smaller space, right? Because I shouldn't do this, right? These steps here, I should just get rid of them and somehow contract certain edges in my space so that I don't go over there. And so this is just something mathematical that happens. Doesn't really help in the algorithm, but usually when you take a polytope and you start contracting edges, you don't get a polytope out. That's just a mathematical fact. In this case, when you have polytopes corresponding to DAGs, or the edges that you kind of put together correspond to DAGs, you'll always get a polytope out. And so our algorithms are actually on a smaller space, namely this thing here, where we contracted certain edges. Okay, so even on this smaller space, so it's not really that you're running an algorithm over the whole permutahedron. You're even running it on a smaller space, which we call a DAG associahedron, and even there it is still consistent. Okay, so you just start somewhere, some random permutation, you look at your neighbors. So here, for example, I'm looking at my neighbors, and I just go to the sparsest one, so in this case, this one. And you just continue on like this. Okay, so very simple algorithm. The problem is just to show that you don't get stuck and that, this is actually a consistent algorithm. Okay, so this is our class of algorithms. I did want to go into it because this is actually how we get consistent algorithms when you also have a mix of interventional data as well. Okay, so let's now get to interventions, which is the main part, what I wanted to get to. So let's first talk about these super invasive kinds of interventions, so these perfect interventions. So here is an example. So say this is my true underlying causal network, and now I'm making an intervention on node 4. So say I'm deleting that gene, so that means I'm setting the value to 0. Well, that means that I'm removing all of the causal effects from anyone coming in, right? Because I'm just setting it to a certain value. I can also do interventions on multiple nodes. So here I'm just removing all of the incoming edges to these two guys. And so now the problem is the following. So I have data, say observational data. So from here, I have data from here. So from this intervention, I have data from here. So from this intervention, I don't know the networks. I want to learn this network. Right? That's the causal inference question. I am, for now, telling you what the interventions are. So I'm assuming I know that here you made an intervention on node 4, and in this kind of data that you got here, you made an interventions on node 3 and 5. Okay, that's a strong assumption, and we'll see that that is actually an assumption that we should get rid of for our kinds of data sets that we're looking at. Okay, so first question is, what are the interventional Markov equivalence classes? Okay, so say I know I have these interventions. Well, how much does that actually cut down the usual Markov equivalence classes that we had in the observational setting? And for perfect interventions, that was answered by Hauser and Bühlmann. So these are people at ETH. And actually, so it's pretty simple. So it says like, well, you basically take your standard Markov equivalence classes, and what you're able to do is you're able to orient edges, in addition, that are adjacent to your interventions. Okay, so you have your Markov equivalence classes, and you're able to orient some more edges, namely the ones that are adjacent to any one of your intervention. Okay, so that's simple. But that was just for perfect interventions, and so it was an open problem. What happens for imperfect interventions? And so what we proved is something that is very surprising for biologists, and actually was surprising to us as well at the beginning, is that actually if you have perfect or imperfect intervention, the amount of causal inference you gain, causal information you gain is the same. So your interventional Markov equivalence classes are just the same, whether you have perfect, the most invasive interventions or not. Okay, so from a biological perspective, it doesn't make any sense to do gene knockout experiments as compared to, for example, okay, so it doesn't make sense to do these invasive gene knockout experiments instead of, just these gene knockdown experiments. Okay, so all these invasive experiments actually don't make any difference in terms of how much causal information you're able to gain out of them. Yeah.
This would be a very naive question, but does that depend on the amount of data you have? The same?
Yes, perfect. So I'm talking, so that's why this is the boundary case. So I'm talking about the Markov equivalence class. So when are you able to get the Markov equivalence class? So this is always when n goes to infinity. Yes. So if you have n going to infinity, and this is perfect. So this is the next question, is to ask, well, if n is finite, right? So I will tell you what kinds of tests we have to do. So we're not using conditional independence tests, but we're using some kind of conditional invariance tests. So how much easier are they to perform, or harder are they to perform for a given sample size? Yeah. So that's a perfect question. Okay, so this is answering the identifiability question. And then, of course, we also have to say something about algorithms. So until, I don't know, some years ago, there was only one algorithm that could deal with interventional data. So this is an interventional adaptation of this score-based approach. Okay, so it's an interventional adaptation of this greedy approach over the Markov equivalence class. So this was, again, proposed by Hauser and Bühlmann. It was conjectured in there that the algorithm is consistent, so that at least it learns the correct interventional equivalence class if you have an infinite amount of data. So we proved it is not. Okay? So even if you have an infinite amount of data, it's in general not going to learn the correct graph. And now comes this thing of why I introduced all of these permutation-type algorithms, because actually it's very easy to add in interventions as well, and you get a provably consistent algorithm under strictly weaker conditions than faithfulness, et cetera. And how do you do it, right? So I'll just tell you in the perfect intervention setting. So I'm in a permutation, and I'm looking at my neighbors. Okay? And I have a certain number of edges associated to me. Now, well, if I have perfect interventional data, I already know that I don't want to walk into a permutation where I suddenly have an edge pointing into an intervention. Okay, that's all we're going to use, but that somehow, that tells you that you can, take away certain search directions, right? You don't want to walk anywhere where suddenly you have some edge pointing into me if I am an intervention. So we're actually performing a greedy search over an even more reduced search space when we have interventional data available. Okay, so that's how we can very easily build these algorithms and actually get provably consistent algorithms that were not available before. And this is to your question. So, how this all works is replacing somehow conditional independence relations with invariance tests. So those are harder to perform. The question is how much more? What is the sample complexity that you need for these kinds of tests, which is all very unknown. OK, so I should end also with showing you how these algorithms work. I don't want to show simulations. For that, you can look into our papers. I always find it a bit useless because you're always showing you're doing better than everyone else. But in simulations, I can do it. We have simulations in there for hundreds of nodes. Okay, on biological data, I can only show it to you for small numbers of nodes because we need to know the underlying truth. Okay, and that's just only, biologists would only agree on that for very small, particularly well-studied networks. Okay, so that's why here, the two examples I'm showing you, you can look at the simulations in our papers, but the two examples I'm going to show you have very few number of nodes just because of that, because I need to know the ground truth. So this is a very well-studied one. So here I'm, basically there are only these algorithms that I can run. So this is the provably inconsistent one from ETH, and this is the perfect version of our algorithm, and this is the one which can, work with general interventions. And this is kind of nice because this is a gold standard data set where they really try to do perfect interventions, and you see that the two algorithms are basically always on top of each other. Okay, so these interventions were really perfect in these kinds of experiments here. And so here is just if you want to recover the edges, and here is if you want to recover them with the directions. Okay, so this works pretty well on these nice data sets. I wanted to show you also how much more work we have to do, and why it is so important to develop algorithms where we might not know the intervention targets. Okay, so this is single cell data. So this is some of the newer data. I didn't put down the date, but this is like data that came out of the Broad with a technique they developed 2.5 years from now. So here you have 8 different interventions, okay? And they, in that paper, looked at these 24 genes. So we're just taking the same, although the data is there for 20,000 genes. So one could actually learn this much larger network. And here we don't know the ground truth, so we're always leaving out one intervention and trying to predict it based on the other. So you leave out one intervention, you use all the other 7 interventions and observational data to try to predict this one intervention. Okay, and you do this for all interventions. So that's what I'm showing you. But this is kind of interesting. So this GIS, which is provably not consistent, is even below the random guessing line. But also what we're doing is not much above the random guessing line. Okay, so this just shows you how much more we have to do. And I think one of the most important points is that we know that for this kind of data, there is a lot of off-target interventions. So even if you tell me that, for example here, that this gene was knocked out, well, in general, also other genes were influenced by this particular intervention. Okay, so that's one of the big problems here. Another one is, although we're using kernel-based independence tests, so this data has a lot of zeros in it. So it's known to be highly zero inflated. And so the question is also, well, can we develop better independence tests that are actually good at detecting deviations from independence in such kind of data, right? So we know that there cannot be any good conditional independence test for any kinds of deviations from independence. So here we would really need to develop a very special conditional independence test that can actually deal with this kind of data. So I think this is another challenge here of how we could get better results, and this is definitely things that we're working on. And maybe also to talk a bit more about the biological setting. So what is it? So here now, okay, so we have a set of algorithms that people can use, but also we have to ask ourselves, what does a practitioner actually want? Usually they don't want to learn a network on 20,000 nodes. They will at the beginning tell you that this is what they want. That I really want to see a network on 20,000 nodes, but nobody is going to be able to interpret the network on 20,000 nodes. So when we discuss these kinds of questions, what we often got out is that, well, what people are really interested in learning differences of networks, for example. So I have, in this case, again, I'm doing it just all in biological language, but I have like a standard healthy cell or a standard healthy state, and I have some abnormal state. And all I care about is usually not learning the full network on both of them, but really knowing what are the different regulations that are happening in both of these networks. Right? All I care about is just learning the differences. And if you care about just learning the differences, then we do have all these algorithms, right? But then don't try to learn both networks separately and then take differences. Right? I mean, this is obvious, but one has to develop algorithms where you just directly try to learn the difference, because that's usually sparse if the two networks are related. So it's much easier to learn a sparser graph than it is to learn two very complicated graphs. And so what we did a lot in recent years is try to actually come up using the kind of methodology that we developed before. Say, well, how can you use it to actually do what people in applications really care about? So how can we, for example, directly learn a difference of graphs? Okay? And here we're applying it to figure out what are the regulatory effects, or what are the real regulatory links that differ between activated T cells and non-activated ones, or here, for example, between what could explain the different survival rates between two different patient cohorts for the same kind of cancer, in this case, ovarian cancer. Okay, and again, you really need to take a causal approach, because this will give you certain conjectured nodes on which you would want to then perform some kinds of interventions and then actually figure out, does this really explain what we're seeing? Does this really explain the two different kinds of effects on these different cohorts? Similarly, if you really care about learning the whole graph, but you have many related data sets, well, don't learn one after the other. Right, learn them all jointly together. So here again, so in our papers, usually we take a high-dimensional statistics approach. So we let the number of variables grow to infinity with the number of samples, and we ask, how much more can you gain by doing this jointly instead of each one separately? And so here we're showing that basically the best is possible, again, with this approach of actually learning the joint permutation and then seeing, learning all the differences between the different graphs. We say that basically the scaling is like the sum of the sample sizes instead of the minimum of each one of the sample sizes. So basically, you can get the most out of your data possible if you do this just jointly. And then I think, again, if you're looking for different problems, I think there are a lot of experimental design questions. Right? So we just started this. So they're a bit different than what people have been studying until now, or often have been studying. So you have a postdoc's life, and that's maybe 4 batches of experiments. Okay, that's not an infinite number of experiments, first of all. And you cannot just perform one sample. You perform one experiment, get the output, and then decide what is your next experiment. Okay, say you have just 4 batches of experiments, and you have all kinds of constraints because you have an amount of money that you're allowed to allocate, right, as everywhere else as well. So sequencing costs. So say you can get the most is n samples. So this should be 100,000 or 1 million. Okay. Now you can distribute them. Usually it's, say, 4 batches. You can distribute them among your batches. Also, preparing libraries for actually performing the experiments costs. Okay. So you can only perform a total of this many experiments. So how many samples should you collect for which experiment, and which experiments do you perform in which one of your batches? Are really important questions that are very important in the setting of genomics, because you have 20,000 different genes that you can knock down and combinations of them. Right? So the search space is huge. So can we actually come up with good strategies for doing this? And I think we did one paper here, but this whole area is really, really wide open. Okay, and I think there are a lot of good ideas that are missing for actually making advancements here. And maybe here it's more important than in other areas to actually just have a constraint on batches. But I think even in, if you want to do personalized advertisements, et cetera, where it's also very easy to perform interventions, I think also there you want to have, you want to be able to say something about the batch setting. You want to be able to say something about where you have constraints on, on your costs of actually performing the experiment, and of your costs of how many users you have for each one of your settings. Okay, so we in the group also take this all the way to early disease diagnostics, which I haven't talked about, so I only talked about this link here, but if you're interested in how to actually take this all the way to medical insights, we have written various reviews, and all of this work wouldn't be possible without an amazing group, and in particular, I will highlight 4 people who were on many of the publications that I showed. So those are 2 PhD students of mine, Yuhao Wang and Karen Dai-Young, and then former undergrad and now master students in my group, Chandler Squirres, a former postdoc, Liam Solus, and obviously a lot of funding. And thank you guys for your.
Thank you, Professor. Now we'll have questions.
So I had a question on interventions when I was hearing your talk. I was just curious, do people apply some rules of selecting what interventions they want to apply to the data? And if you do so, like maybe depending on the sort of causal questions that they want to answer, do you incur any sort of selection bias like in the analysis that follows if you sort of choose the interventions that you decided to pursue?
So there are 2 different, so until now, so there is a question of like perfect interventions or not. Until now, they tried to do perfect because they thought that you would get more information out of them. So now probably imperfect should be about the same. But then, and the other question is all these experimental design questions. But there we mainly, and this came up also in Nathan's talk this morning, I think, so how you would, since we're allowed to choose the interventions, right? So until now, actually, so they're super interested. This is like at the Bro David Regev, who did all of these experiments here. They're super interested in strategies. So for now, how these interventions work is they have viruses that contain, so you have a strain of viruses that can do this gene knockout, strain of viruses that can do this one, this one, this one. Now you infect your cells, okay? And so a combination of viruses can infect the cell, and hence that means that all this combination of genes will be knocked out. So for now, they just do this randomly, okay? So they'll do 1, 1, 1, 1, but they can also do any combination, and they would really like to know, right? What is the percentage of these guys that I should actually put to my cells? Also, how many of them should I put? Because if you put less, you get more observational data, so no intervention at all. If you put more, you'll get, more that actually show an intervention. So, but until now, they're doing it randomly. Okay. So this is randomly when you don't have anything that you're studying. You really just want to learn the underlying gene regulatory network.
So maybe it could be a naive question, maybe I'm getting it wrong, but do you maybe select these interventions based on a certain set of causal questions that you want to answer? Say, I'm interested in something, I try intervention A. I see that doesn't give me much information about the question that I'm trying to answer. So I try another intervention, and then I go on. So
yes, okay, so these are different kinds of questions. So here it's a question about learning the full underlying network. Okay? But if you have a particular area in your network that you want to really learn about the most, right? Then I think this is how we framed this. This experimental design question. So, so how do you want to choose your interventions? Well, you want to choose them so that it reduces the uncertainty over the network structure. So now you can then ask, well, okay, I want to reduce the uncertainty over the network structure in this particular area of the network. Okay, and so we're taking a Bayesian approach. So we're saying, okay, I should add more interventions here because my expected gain in terms of uncertainty reduction is going to be this much. So these are the experimental design questions, which I think still need a huge amount of work. But that's how we have looked at them in terms of uncertainty reduction that you can get from a particular intervention. And then you can start looking at particular areas in your network.
Are there any further questions? Okay. Well, let's thank you, Professor, again, and take a short break and reconvene at 4.